So hello everyone and welcome to this journal club from the Atheronetcos Action. Today we will be talking about transcriptomic changes in the atheroma and how this can be leveraged for a better understanding of risk stratification in atherosclerotic patients. We will be discussing a recent paper published from groups in Karolinska, and today we have the pleasure of introduce our two speakers, Dr. Narayanan, which is a research engineer with expertise in bioinformatics and clinical data from atherosclerotic patients, which is currently developing computational frameworks in cancer research. Then he would be presenting together with Ms. Vuckovic, which is a PhD student from Karolinska as well, that after completing her studies, moving across Europe from Netherlands to Germany to Sweden, then decided to take on a PhD project working on smooth muscle cell biology and cardiovascular disease. The floor is yours, so please go ahead.
Thank you so much for that nice introduction, and on behalf of both Sampath and myself, thank you for the opportunity. It's a very big pleasure to be presenting in this nice network. So I hope we can have some nice discussions.
Happy to have you guys.
We recently published a paper in the European Heart Journal, and the title reads, Atheroma Transcriptomics Identifies ARNTL as a smooth muscle cell regulator and with clinical and genetic data improves risk stratification. So before we go on to the actual science, Sampath, can you go to the next slide? We have no disclosures. And I will probably go over some points that are familiar to all, but just a quick reminder maybe that single-cell RNA sequencing has in recent years become a very important method in the space of vascular disease research. And in the context of atherosclerosis, it's particularly important, being that the different cell types present in the plaque are not the same in different plaques. Namely, the proportion of these different cell types differ from person to person. So we are in particular focusing on mesenchymal cells, and in particular in that space on smooth muscle cells in our group, and this is of relevance for this project as well. So the smooth muscle cells migrate from the media where they reside in normal situations into the plaque and participate in the plaque development and atherosclerosis progression, eventually also impacting the outcome of the disease and the plaque profile. At least that is the idea that the field is at right now. So the smooth muscle cells in media are contractile, as their main role is to regulate the diameter of the vessel and to provide structural integrity. However, during atherosclerosis, they lose their contractile properties and acquire both markers and functions related to many other cell types. And these other cell types include macrophage-like cells, osteoblast-like cells, myofibroblast-like cells, and so on. We can go to the next slide. So the field has recently been exploring these smooth muscle cells in more detail in atherosclerosis, but genome-wide association studies have so far confirmed few variants in coronary artery disease, or CAD, that have direct implications for smooth muscle cell function. You can go forward, Sampath. But we think that the genetics in the population and the clinical risk factors may directly affect the distribution of smooth muscle cell subphenotypes in plaques. So what does this mean? That in some people with certain genetic variants and certain risk factors, the smooth muscle cells may be stimulated towards a macrophage-like phenotype. And we know from previous studies, namely mouse studies, that in increased presence of macrophage-like smooth muscle cells associates with increased inflammation and increased risk for rupture. On the other hand, some patients might have smooth muscle cells that are dominantly transdifferentiating into a myofibroblast-like cell type or a synthetic cell type. And these will produce a lot of extracellular matrix that will thicken the fibrous cap and thus make the plaque more stable and less likely to rupture. We can go to the next slide. And indeed, such changes in the distribution of smooth muscle cell subphenotypes may lead to differences in plaque stability. Eventually, this might directly impact the risk for stroke and myocardial infarction. We can go to the next one. Yep, thank you. So that led us to formulate the hypothesis that CAD genetics and clinical risk factors may influence smooth muscle cell subphenotypes in plaques. And this is the central research question of this project and some others that we are running in the group. So the resource for exploring this research question is the BIKE Biobank. And BIKE is a shorthand for Biobank of Karolinska Endarterectomies. This biobank has been established more than 20 years ago and currently collects samples from over 1,300 patients. And the really nice thing about this biobank is we don't only have many samples, but we also have a lot of data associated with the patients that have donated their biomaterial. So being that the biobank was established in 2002, we have also gathered a lot of follow-up information from the patients that have donated the plaques. So this collection of biomaterial, together with the clinical, epidemiological, and other data that we collect from these patients, provides a really nice resource to explore genetics, risk factors, the effects of medication on mesenchymal cell phenotypes in the plaque. So the aims of the study were thus to correlate smooth muscle cell subphenotypes to patient clinical parameters, to associate CAD GWAS variants to these SMC subphenotypes, and therefore understand if there are any novel SMC-specific gene signatures that we could then decipher the role of in plaque biology. We can go forward. And the approach that we took to achieve these aims was deconvolution of bulk plaques from BIKE. So we started with 127 carotid plaques from BIKE that were subjected to microarray to evaluate their gene expression profile. On the other hand, we took data from the Stanford Biobank where they had 5 coronary plaques that were subjected to single-cell RNA sequencing. And this was then transformed into a single-cell gene expression signature matrix. And when this signature matrix was combined with the bulk RNA sequencing data from BIKE, we could come up with an imputed profile for each of these BIKE plaques, describing their relative cell fractions. And this deconvolution was performed with CyberSort, which is already an established software used for these purposes. And it was very important to stratify the bulk RNA sequencing data into cell fractions, because this can then be associated with symptomatology in BIKE, the sex of the patient from which the plaque was taken, as well as medication. And BIKE, being the resource that it is, allows for all of these different associations, which was really inspiring to work on. Next slide, please. So moving into the results, the distribution of cell fractions in plaques was obtained, as you can see here. And we already see at this stage some interesting data relating to symptomatology. If you take a closer look, we can, for example, see that symptomatic patients have an increased presence of macrophages in their plaques, which would be expected. And another interesting finding pertains to parasites that are also higher in symptomatic patients. And this may be reminiscent of the fact that symptomatic patients likely have more neovessels in the plaque, which then again associate with intraplaque hemorrhage. But we would particularly like to focus on smooth muscle cells, as I mentioned before. So if we go to the next slide, we can take a closer look at what is happening with these cells. So we can see that for SMCs1 and modulated SMCs, we have a decreased presence of these cell populations in symptomatic plaques. And what are these different SMCs that we see here on the graph? Well, SMC1 is a more classical smooth muscle cell phenotype with expression of genes relating to contractility, and on the other hand, modulated smooth muscle cells express low levels of these smooth muscle cell markers, and on the other hand, express some other markers that we associate with usually with other cell types. And SMC2 is a fraction in between these two. But what is interesting is that in symptomatic patients, there are less of all of these smooth muscle cell fractions, which is something that we would also anticipate from previous research done in this field. Next slide, please. So then we used this data coming from the stratification of cell types in bike plaques, and we performed unsupervised clustering. And interestingly, we observe 3 separate clusters that almost don't overlap at all. And these clusters were interesting from a perspective of follow-up data. So when we looked at cumulative ischemic stroke events, we see that cluster 3 confers the most risk. And this is one of the first attempts to verify that the cell fractions in plaques actually have something to do with the prognosis of the disease. So we found this very exciting. We can go to the next slide. So let's look at this in a little bit more detail. When we look at the different cell fraction distribution, it provides granularity for this risk stratification. So as I mentioned, cluster number 3 conferred the most risk. And if we take a look at the first 3 panels here, we can see that smooth muscle cell fractions are the lowest in this cluster, and that agrees with the symptomatology association that we saw a few slides before. Interestingly, the inflammatory cell populations are highest in this cluster, while endothelial cells are also high. And here I would like to compare cluster 1 and cluster 3. Both of these are relatively similar when you look at the first 2 rows. However, when it comes to endothelial cells, here they are higher in cluster number 3. And we think that this is maybe because the cluster 3 has increased new vessel formation, which then renders the plaques more vulnerable for intraplaque hemorrhage and therefore also for rupture. On the other side of the spectrum is cluster number 2, which has a lot of smooth muscle cell presence, not a lot of parasites, which are again associated, as I mentioned, with new vessel formation, and less immune cells present. Next slide, please. So we have already now established that the cell fractions have something to do with plaque biology and also with the outcome of the disease. However, does this have anything to do with the fact that we have deconvolved bike plaques, which are carotid plaques with the coronary single cell sequencing data sets? This was a question that was posed by one of the reviewers, and Sampath will talk about that more in a minute, but we wanted to verify basically the deconvolution results that we have observed and shown in the previous slides. So we have verified that with the carotid single cell RNA sequencing data set, we observe relatively the same results. And furthermore, if we go to the next slide, we can observe that the similar clustering occurs also in this case. So we see again 3 clusters that don't overlap, and one of the clusters is highlighted as the most associated with risk. And in this case, we see a difference when we look at cumulative myocardial infarction events. So here I would like to close my part and ask Sampath to continue. Thank you.
Thank you, Sofia. So, so one of the aims of the project, as Sofia mentioned, was to understand if how the patient genetics, the cardiovascular patient genetics, if and how they have any influence on the different plaque subphenotypes of mesenchymal cells, especially smooth muscle cells. So to do this, what we did, we took a unique approach wherein we collected a list of already published risk variants in cardiovascular disease from literature. And we associated these variants with the mesenchymal cell fractions that we deconvoluted And the highly correlated variants with the mesenchymal cell fractions, they were called the cell fraction quantitative trait loci. These variants represented the influence of cardiovascular genetics on the cell fractions. But we also wanted to know if there are any particular set of genes that influence which mediate the influence of genetics on the cell fractions, the mesenchymal cell fractions. So to investigate this, we performed EQTL analysis on the cell fraction QTLs, and we ended up with a list of genes that are that had significant correlation with the uh, with, we ended up with a list of variants that had significant correlation with the genes that they were mapped to. And if you look at the genes at the top of the list, we found that several variants were mapped to one particular gene. And these variants were present at either upstream or at the regulatory regions of this gene, ARNTL. So we wanted to know how ARNTL is involved in regulating the different smooth muscle cell subphenotypes in the plaque. But before that, we also wanted to know the influence of the importance of these variants in terms of the disease. So we investigated the association of these variants, the top variants, in a cohort which had patients with at least three cardiovascular risk factors. And there we found a significant association between some of the top variants from our analysis to the presence of carotid plaque in the patients. So this established an important link between the presence of these variants and the disease itself. So that is an important finding. But as I said, we also wanted to see what is the link between the variants and the gene that the variants map to, and how this gene relates to the function of smooth muscle cells in various subphenotypes. So this gene that these variants are present in, ARNTL, and the protein that it codes to, which is BMAL1, is an important transcription factor and a master regulator of circadian rhythm in mammalian cells. This protein, along with another protein called clock, they form a complex which binds to DNA regions in the DNA called E boxes, and they regulate the transcription of genes that play important roles in fundamental cellular processes such as proliferation, apoptosis, cell differentiation, and metabolism. So then we investigated how the gene expression of this gene is regulated by the SNPs that we found in this gene, and we found that the risk allele of all these variants were associated with decreased expression of ARNTL in bike plaques. This means that the presence of the variants are associated with a decreased expression of the ARNTL in the carotid plaques. And we also found that the actual gene expression of ARNTL itself was downregulated in the carotid plaques, and we were also able to identify that BMAL1 was present in the areas of the plaque where there is an increased abundance of smooth muscle cells, which indicates that the role of ARNTL might be important in smooth muscle cells. So to understand the role of ARNTL in smooth muscle cells, we turned to in vitro cell culture, where we used human primary smooth muscle cells to understand the role of ARNTL. So we silenced ARNTL in smooth muscle cells, And we can see that the gene expression of ARNTL was increased in response to PDGF-BB, which is a potent stimulator of smooth muscle cells. But when ARNTL was silenced in these cells, then this increase was abrogated. And silencing ARNTL also resulted in a decrease in proliferation of these cells and an increase in the contractile gene expression of smooth muscle cells, as you can see from this graph. In addition to a decrease in proliferation, we also notice that these cells were positive for beta galactosidase, and also the gene expression of certain genes that are involved in senescence were also significantly higher in the cells where ARNTL was silenced. So with this, we concluded that ARNTL regulates the proliferation and senescence in smooth muscle cells. So to conclude, we used a unique bioinformatic approach to establish a relationship between the different smooth muscle cell subphenotypes and the clinical symptoms of atherosclerotic patients. So as we saw, some of the variants that are causal for cardiovascular disease seem to influence the subphenotypes of smooth muscle cells in the plaque. And this is important because there is a lot of literature about variants that have causal relationship in cardiovascular disease through either inflammation or which affect the lipid either inflammation or the lipids. So this is one of the few studies that focused on how some of the cardiovascular variants might affect smooth muscle cells function. So this gives another avenue of approaching treatment strategies by targeting smooth muscle cell function in in patients with cardiovascular disease. So back to the study, we identified several variants in many genes, but a lot of variants in ARNTL that may have functional implications in smooth muscle cells. And the risk alleles in the ARNTL variants reduce the gene expression of ARNTL in carotid plaques. And we also showed that the effect of ARNTL in smooth muscle cells is through control of proliferation and senescence in plaques. So to summarize the whole study, we approached the study through different questions. For example, first we asked how the genetics in terms of cardiovascular variants influence the different distribution of carotid plaques. And in order to find this, we had to sort of decompose the whole plaque into different cell fractions. And this we made use of the bioinformatic approach of deconvolution to do this, and we obtained cell fractions. And then we also correlated the different CAD variants with the cell fractions to obtain what we call cell fraction QTLs. And we wanted to also know how if there are any genes that mediate the effect of genetics on the different distribution of these smooth muscle cell fractions within the plaque. So for that, we performed eQTL analysis of the cell fraction variants, and we ended up with a list of variants that have significant effects on various genes. And since a lot of variants mapped to ARNTL, we wanted to we asked the question if ARNTL had to do anything with the mesenchymal cell function or smooth muscle cell function. So then we turned to in vitro data to perturb the gene expression of ARNTL to study the function, and we ended up with the finding that ARNTL affects the proliferation and senescence of smooth muscle cells. So that is about the study. Finally, I'd like to acknowledge our the group leaders, Associate Professor Ljubica Matic and Professor Ulf Hedin, who have been, whose constant support throughout the project was very important for us. And also our collaborators from Stanford and University of North Carolina, who provided us with the important single cell data that we could use for deconvolution. And I'd also like to thank all of our collaborators, who played a very important role later in the study when during the revision process and so on. And I'd also like to thank Sophia for her invaluable contribution for this work. Um, I'd like to discuss a little bit more about how the revision process went, and this might benefit a lot of early career researchers in the audience as well. So, the revision process, the whole process took about close to a year, with 4 revisions, and we had like 4 reviewers, looking at our work, and there were important learnings for us, and there were things that were very rewarding in the revision process, and things that were challenging, and so we learned a lot from the review process itself. So for example, the most rewarding part of this is, as Sophia mentioned, when we started this project, the single cell data from the coronary plaques were the only data, that reliable single cell data, that was available at the time. So then we used that to deconvolute carotid plaques. But then by the time we submitted the manuscript, uh, and and the reviewers were right to question if there was any vascular differences between the vascular beds from coronary plaques to carotid plaques that we were trying to deconvolute. So then, so this gave us an opportunity to explore the deconvolution in other publicly available data sets, to deconvolute other carotid plaques to see if this is consistent with what we did with our bike plaques, and we did just that. And then by the time we submitted the manuscript, there was a lot of literature on single cell. There was a lot of single cell data from carotid plaques as well. So we were able to get our hands on one of the carotid single cell sequencing data, and we used this to deconvolute our bike plaques. And that was also that also agreed with what we had done using coronary plaques. So all of these validated our deconvolution approach, which was the sort of Achilles heel on the whole study, because uh, obtaining the cell fractions from the plaque was very important to correlate that with any clinical parameters, and also with the genetics and so on. So that was very rewarding for us, and that reinforced the very idea of the project. And one of the other things that was challenging was that um, this was largely, the whole project largely focused on the clinical data and the bioinformatic aspects of deconvolution. So what happened was, the in vitro data happened as a sort of a proof of concept of what we obtained from the bioinformatic analysis and the clinical analysis. So then we tend to do this a lot. We tend to put our findings out there, but what is more important is the rigorousness and the robustness of this approach. So when we submitted, we got a lot of questions about the in vitro data that we needed to address, such as quantification of the Western blot results, for example. And the we did initially, we did not include a proper positive or negative control for the BMAL1 protein, which is the main protein of study in the project. So these were all, the reviewers pushed us quite a bit on these things, and those things, the main learning is that even if it is a proof of concept, or if it is one part of the manuscript, we need to so if we had done, for example, if we had included the positive and negative controls very early on from the beginning, then we could have avoided this, and that is the right way of doing things as well. So that's very important to keep in mind. The other thing, I think this can also be said of most of the basic biology researchers, is that we tend to look over the statistical rigorousness of our data. In our case, what happened was, for example, when you have multiple statistical tests, then it is very important to perform, for example, multiple comparisons and correct the data for multiple comparisons. And the way we expressed certain terms, certain biological concepts, were at odds with the way statisticians perceive those things. And so this was expressed in terms, in figures as well, and also in terms of how we describe the results. And these were also interesting for us to correct and also for us to reflect back on. And so those are the things that we also always need to keep in mind, that while the data is important, the statistical rigorousness of the data is as much important as the data itself. So those were the main takeaways for us from our revision process. And while it was a bit challenging, it was also rewarding. So that in the end, we were happy with the whole process. So that's all we have, and we'd be very, and as Sophia said, we are very happy that well, we are very thankful that Athronet actually approached us to present this research. Thanks to you guys. And yeah, we'd be happy to take any questions about the study if you guys have any. Thank you.
Fantastic presentation. Thank you both for sharing this huge amount of work that has been done to get this good paper published, and also for sharing your thoughts about the revision process, because that's very important. Like, great papers need great work also, behind the scenes, and not only until submission, but after. Like, revision process can be lengthy, but makes the paper better, as you were saying. I will let Melody and Chris take over for the Q&A session. So Mel, please feel free to jump in.
Thank you both for an impressive presentation and an amazing work that has taken so long in the revision process. I've been behind the scenes a little bit with the struggles of the revision, but I mean, I'm sure you're very excited to have it out, and it's very impactful publication. I think we have a question from the discussion from the chat here from KM. Do you have a hypothesis on how ARNTL expression might be associated with adipogenesis from figure 5F? Karin is asking this question.
So maybe I can start by saying that I don't think we know, and we didn't really explore this in much detail, but I secretly Googled this quickly when I saw your question, and it seems that there is already some research on that. So there is a paper that describes its role in cellular hypertrophy by increasing insulin sensitivity via FGF21, and that's just from a quick search. So I'm sure there is more out there. But as Sampath said, the role of ARNTL in this process and related to smooth muscle cells in particular is important, and we suggest it as a subject of further study. But actually, the core of the entire paper is the proof that certification of cell fractions from the plaque is really important. And not only that it can deliver molecular players that are interesting to study further, such as ARNTL, but it can also provide a way to stratify the patients, really strengthening the idea that atherosclerosis can be interesting for approaches that are more personalized for the patients. So I know I didn't answer your question, but I hope it provided at least some perspective on how we thought about this particular target in the context of this project.
Sorry, I was trying to look what figure 5F was, so that's why the delay from my part. But the thing to remember is, first of all, we did not explore the role of ARNTL in adipogenesis. That's first. But then ARNTL is also a very, very fundamental gene, which has its hands on a lot of fundamental process. So I'm sure that it will be involved although we don't know, it will be involved in a lot of processes, which might be the case with adipogenesis as well. But as Sophia said, I think this gives a method of understanding how the genetics causes certain plaques to be a certain way in terms of the terms of the distribution of cell fractions. Yeah. So thanks for the question.
You can feel free to unmute yourself if you also want to ask the question live to Sofija and Sampath.
I actually had a question, if I may.
Please.
Can you hear me?
Yes.
Yes.
Sorry. Okay, I'm having some issues with my microphone. A little bit with respect to the question, just with respect to BMAL1, I know that it's a protein of the circadian cycle, and upstream there are the ROR family of proteins, which are also involved in the immune response. Given that you guys were looking at plaques, do you have any insights on? I'm not sure if I missed this in your presentation. Any insights on the immune cells in this context?
Maybe I can start. So it's very true because, for example, we did not have, at the time we submitted the manuscript, we did not have a proper single cell data to look at the distribution of gene expression. So we made use of publicly available data sets to see how ARNTL is distributed in different cell clusters. And apart from smooth muscle cells, for example, macrophages are one of the cell types where there is a considerable expression of ARNTL. So as you said, as much as it might play a role in smooth muscle cell regulation, it definitely has important regulation to play in immune regulation as well, and that might be very important in the plaques as well.
Yeah, even with regards to crosstalk between the immune cells and smooth muscle cells, perhaps.
Probably, yeah, definitely.
Okay. Thank you.
Any more questions from the audience? Feel free to interact with Sofija and Sampath, and you can unmute yourselves.
So while we wait, maybe one from my side. As you guys mentioned, the validation in the end on in vitro validation on RNTL modulation was just as a proof of concept. So you just guys went and used an siRNA approach. But I was wondering whether it's something you're interested in and expanding on to know how the different SNPs may affect and small muscle cell behavior in vitro, perhaps using a CRISPR-Cas9 approach rather than just silencing the gene as it is. Is this something you guys have done, tried, or like are working on?
Very fair question. So yeah, I mean, to start off, that's what we tried to explore, and the approach with the siRNAs happened because primary smooth muscle cells are very difficult to use in a CRISPR-Cas9 setting.
I've been there. Yeah.
And yeah, so this is a proxy for it's not exactly that would have been beautiful, if we had knocked out one of the variants, that would have been perfect. But we couldn't do it in this setting. And using a cell line would not actually replicate the primary, how the cell line would behave. So those are technical limitations that we had. So we haven't planned anything like that because of the of the technical limitation. So we had to settle for silencing the gene expression instead.
Thank you for sharing. I reckon my real question was whether you guys have the tools, and if so, please share them with us, because
No.
we all know how difficult it is to do this on primary smooth muscle cells.
I mean, we experience the same difficulties.
Thank you for sharing that.
If I can add, I believe that Minna Kaikkonen's lab in Finland has some experience with this system. And we briefly discussed an idea maybe of starting a collaboration and sharing some methods in the context of this project, but then we prioritized publishing it instead of expanding it further and doing the approach that you suggested. So I think that she might be able to help in this context. So
Definitely,
enough.
that might be interesting.
We also did have some success, but let's say when we start, I mean, just my small experience, we did this a few days ago, and like the tools that were available back then were not like ideally, let's say, not ideal to try and do like single nucleotide mutation with CRISPR, while now maybe like, yeah, the technology is out there, but yeah, maybe for those starting a project rather than wrapping up one. Thank you both. Any additional comments from your side, Mel?
Yeah, I could maybe ask more of a philosophical question about what would you think would be the follow-up work from here? Where would you like to take this work further? What would be the next step for this study?
Well, I think there's a lot of interesting directions when it comes to smooth muscle cells and atherosclerosis, and anything that we find out is potentially beneficial because the field is so rich right now, and a lot of people are working on it. I think there's also a lot to uncover. So, as you know very well, Mel, we have in our lab some efforts to explore the role of smooth muscle cell transdifferentiation in more detail. So we could always go back to some of the targets we found in this project, not only ARNTL, but others that were ranked highly on this in this table that Sampath showed, and evaluate how they behave in the different transdifferentiation profiles of smooth muscle cells. So that's the one direction that I can think of on the top of my head. A very biased answer from my own research.
No,
Nice.
also, if I may add, the list that what we performed, there are a lot of variants that are not characterized as yet. What we cannot find in other published research. So it's a very difficult work to to characterize the function of those genes, but that might give us a novel angle on what they might be doing in smooth muscle cells. So that might be one of the potential future works as well.
Very nice.
Thank you for sharing. Probably a final comment, unless someone else has questions from the audience, is building on what Sofia was saying. You guys have done a fantastic work trying to correlate genetics with cell fractions, but transcriptomic, like these new technologies, transcriptomics to start with, are letting us understand that there is much more behind the cell types that we know about in disease, especially, the transdifferentiation potential of smooth muscle cells, endothelial cells, and so on. So I was wondering whether there is space for these tools and the cell fraction characterization and the signatures we use for cell fraction to be improved. So potentially giving us an even better resolution of how we can stratify risk in patients, because we are doing this basically, if I understood this correctly, on what is known so far about the transdifferentiation potential of cells and so on.
Certainly, I mean the number, the more patients we profile in terms of single cells, and the more data that we mine, the more we know about the heterogeneity of these cells in the patients. And I wouldn't say if that might give us exactly that is linearly correlated with the risk in the patients.
Right.
But for example, I can show you already this one. Because the single cell data from the carotid plaques that we got later for the revision, that already had a lot of phenotypes, a lot of granularity on the phenotypes compared to so if you look, there is a lot of mesenchymal cell types and macrophage cell types. And I don't know if that is the reason why you have a better separation and a better risk stratification, but it might be the case, and someone has to dedicate some more time and space, time and resources in looking at that, and that might be one of the future work as well. So the more granularity you provide in terms of plaque biology, the more granularity you provide in terms of risk stratification as well. We don't know, but the plaque biology plays an important role in how the plaque might behave in terms of symptoms.
Definitely. Yeah.
So yeah.
And to add another point, I think a direction that the field is expanding in is also integrating the spatial component.
Oh yeah.
So I find it
Absolutely,
very it's going to, well, I don't know if I'm right, but it might be that smooth muscle cell location will be very strongly associated with symptomatology and disease outcome. And if we identify different markers that play a role in smooth muscle cell function, such as ARNTL, and if we understand the spatial relevance of this particular marker and also the transdifferentiation phenotypes more generally, I think this is going to provide an additional level of understanding of the plaque biology and the importance in the cell types that are involved in the disease. So that is coming, I would say.
absolutely. Very exciting times ahead. Like, to know more in time and space will definitely provide more information for us. Mel, what do you think? Shall we thank our presenters and wrap up the session?
Yeah, I guess so. I think if no one else has any comments or questions, we can we can close the session. I wanted to also say that Sampath had a good point about looking at the cells as a spectrum and not really as a phenotype. I think I really like.
Yeah. Very good point. Yeah. So thank you so much, everyone, for joining today, and thank you to our presenters for sharing this fantastic work with us. That's all for today. Hope to see you all in our next session next month. We will be providing information about that soon. Thank you, everyone.
Thank you all.
Thank you.
Thank
Thank you so much.
Bye-bye.
Bye.
Bye, everyone.
Bye-bye. Thank you guys for organizing and for having us.
Thank you so much.
Thank you so much for joining.
Thank you guys for presenting your work.
It was a pleasure.
That
Bye-bye.
was fantastic. Yeah.
Thank you,
Bye.
Sofija.
How are we doing, guys?
Good. How are you?
Not bad. It was a very nice session. I'm very happy
super
with
Yeah,
our
it's good.
interaction
It's really
lately.
good paper. Yeah, I wish we had more participants, but
Yeah, it was not great. I mean, yeah, but we at least